Now we see that there are some differences in these birds. They look distinctive, they sound distinctive. So what are genetic differences that would lead to this? So we're going to step back and examine the whole principle of genetic differences, and then we're going to apply it to the blue-winged and golden-winged warbler. So genes, which you're familiar with this term, are what lead to different appearance and characteristics of organisms. And you've heard the idea of genotype leading to phenotype. So genotype is the genes that an organism has, and phenotype is the way that the organism appears. But it's a little more complex than that. Genes code for different proteins, and those proteins lead to the differences in phenotypes. So we want to think about both genes as well as the proteins that they code for. So this relationship is called the fundamental concept of molecular biology, where we start with a gene that's located on DNA, and we go from DNA, which is made up of nucleic acids, we abbreviate those A, C, G, and T, to RNA, which is also made up of nucleic acids, in this case ribonucleic acids, whereas DNA is made up of deoxyribonucleic acids, but similar structures. And then you have proteins, and proteins are made up of amino acids, and we abbreviate those with three-letter codes, and they play a role in our body of being structural things or chemical enzymes that help lead chemical processes in the body. DNA to RNA to proteins occurs through two processes. The first process is called transcription. And transcription occurs when, using a DNA template, a new molecule is made that is the RNA, and that RNA molecule then leaves the nucleus and can be used to create proteins. Proteins are created through a process called translation, where the instructions are read off of the strand of RNA, and for each 3 nucleic acids on the strand of RNA, 1 amino acid is put into a new strand of chemical called the protein. So it's a 3-to-1 ratio, and so in the end you have still all 3 molecules. Nothing happens to the DNA. Then you have the RNA molecule that kind of allows you to transmit or transfer those instructions, and then you end up with the protein, and the protein is what does something or gives a certain appearance to an organism. All right, so we have those. And how do you remember transcription versus translation? So these are things that you'll get more into depth in LB 145, but I like to use a very simple technique to remember this difference, and that's translation has the word late in it, and it occurs later in the process. So the first thing that happens is transcription, and then translation happens later. Now transcription is when you take one language and you transcribe it into something very similar, essentially in the same language, whereas translation is where you go from one language to another. And if you think about that, deoxyribonucleic acids to ribonucleic acids, those are very similar. Ribonucleic acids to amino acids are very different, so there you have the translation occur. Now, DNA is stored information, RNA is the temporary instructions, and proteins are the functional products. So let's look at some things that we can think of as where there's a lot of stored information. Well, Wikipedia is a place that there's a lot of stored information. We used to have encyclopedias we thought of as that stored information. So the DNA is like the Wikipedia for an organism. It has all of that information about how to build the organism. Now, to make that information usable, it is turned into temporary instructions, kind of similar to a blueprint. And those temporary instructions can be exported out of the nucleus. And those then can be turned into functional products like tools in our analogy here. Now, if we look at the way we write these things, DNA we write as a sequence of A, C's, G's, and T's. And so here we have a strand, TCT, GAC, CC, TC, AAG. And that then would get transcribed into an RNA strand. And the RNA strand, you can see the letters are different because they're corresponding letters to the DNA strand. But you have one letter for each of the DNA letters. One base pair for, or one base for each of the bases in the DNA. From there, you take 3-letter chunks of the RNA and match those to a particular protein. In this case, there are 12 RNA bases, and that leads then to 4 protein amino acids. All right. Now, what happens when you have a mutation? Let's say that C gets changed to a G in a point mutation here in our DNA. Well, then when that gets transcribed, that leads to a difference in the RNA sequence, and that can lead to a difference in the protein sequence. Here we see at the 3rd position, instead of glycine in the top protein, we have arginine in the bottom protein. So you can have that result in a change in the amino acid sequence, all right? And that makes slightly different instructions. That leads to slightly different blueprint. And the question is, does that lead to a very different tool? Or does it really just lead to a slightly different tool where in typical cases you have a binding site on a protein that allows a chemical process to happen, and that binding site might change its shape or its size based on the mutation that occurs. So this gives us an idea of how we have DNA, RNA, and proteins. And let's talk about some of the key terms then that we use with these. So one is the term gene. And gene is a segment of the DNA that codes for an RNA strand. And in humans, we have 23,500 different genes. Okay? The RNA strand that we're most concerned about here is called mRNA, and these are messenger RNA strands, and these are the ones that get translated into proteins. And again, proteins are the thing that make organisms different, have different appearances. And then you'll also see the term genome from time to time, and the genome is the entire genetic makeup of an organism, including all of its chromosomes. So, what do genes do? If we go back to our DNA to RNA to proteins, genes are segments of DNA made up of A, C, T, and G bases, and they provide the instructions for making proteins. So, you have differences in your genetic makeup, and those are responsible for some of the differences you see in the phenotypes of organisms because they lead to the production of different proteins. Different versions of genes are called alleles, and then different alleles would lead to different proteins being made. Okay. And again, we think about this, we think about the genotype of an organism, that's the genes that it has on its DNA, and the phenotype of the organism is a combination of the proteins that it has as well as some of the environmental factors that lead to it having slight differences. What do proteins do? Proteins have 2 major roles in the body. The first is as enzymes. Enzymes are things that convert one molecule to a different molecule, or they help assemble molecules or break down molecules. A really important enzyme, for example, is insulin. Okay, this is a very important protein, and it converts glucose to glycogen, and you can see the structure of glycogen shown here in the upper right. And that glycogen is a way that glucose can be stored, where hundreds upon hundreds of glucose molecules can be stored in one location. You can also have an enzyme like the MC1R protein. What does that do? MC1R protein triggers a reaction to produce melanin, eumelanin in particular, which is a black pigment. And if we think about that black chin patch on the golden-winged warbler, we'll see that it probably needs there to be melanin produced in that location to create that black pigment. Proteins are also structural. One of the key ones is collagen, but they make up physical structures of a membrane or inside a cell or between cells. And collagen, for example, is the primary building block for skin, for muscles, and a lot of connective tissues. So, with these two warblers, we would expect there to be genetic differences that lead to the difference in appearance of the two species. So we would expect there to be some sort of allele for golden-winged warblers that leads to production of a yellow pigment, and that the blue-winged warbler would have an allele that leads to the production of white pigment in the wing. So in the golden-winged warbler, we see golden in the wing because of a particular allele, and here we see white in the wing because of a certain allele. The golden-winged warbler should have an allele that leads to producing that black chin, and that should be absent in the blue-winged warbler. And the blue-winged warbler has an allele that leads to producing that black line through its eye. All right. So those would be different alleles you might expect to find in it. It's not always that simple, but if we follow kind of the straightforward idea of allele to protein, then there should be an allele for each of those differences.